Okay, well, welcome to one of the highly sought after PowerPoints about cell signaling. So remember that there is a lot of cell signaling happening all over in all of your cells all of the time. There are a lot of molecular switches that can turn proteins either on or off. Two of those switches that you need to know are phosphorylation, which are driven by kinases. So if you add a phosphate, the enzyme is called a kinase. The enzyme that removes a phosphate is called a phosphatase. Phosphorylation can be on or off depending whether it's phosphorylated or not. It's completely protein dependent. GTPases, on the other hand, are proteins that bind either GTP to turn it on or GDP to turn it off. An example of that is the RAS oncogene. In the RAS oncogene, mutations cause it always to be GTP bound and always on, signaling constant cell division. So in cell signals, we are using cells communicating through chemical signals, usually proteins or small molecules, where we have a signaling cell that is giving off that signal and a target cell that has a particular receptor. That is the cell that's going to respond. If it doesn't have a receptor, it's not going to respond. So when we're talking about cell signaling, we're talking about ligands and receptors. A ligand is the signal, and the receptor is the thing that is going to bind that signal. So ligand binding changes the shape of the receptor. So remember, ligand binding, receptor changing shape, leading to some sort of signal transduction and a cellular response. You can have endocrine signaling, meaning it's going through the blood, it's very far away. Paracrine, meaning nearby. Neuronal, meaning it is very, very close through a synaptic terminal. And then we can have contact-dependent signaling, where proteins actually are touching. There's one other form called autocrine, where cells are signaling to themselves. Earl W. Sutherland Jr. was the first to discover about cell signaling. He studied epinephrine or adrenaline. And what he saw is that we knew at this point that adrenaline or epinephrine was a hormone secreted in the blood by the adrenal glands, which are the little organs that sit on what are they called? The little glands, that sit on top of the kidneys, like little yarmulkes. And this signal causes glycogen to break down into glucose, which helps the fight or flight response. So what he did is he added epinephrine directly to glycogen, and you do not get a response. So it turns out that in order for epinephrine to cause this signal, it has to have an intact cell with an intact receptor and all of the signaling molecules. So there's 3 stages of all cell signaling. Ligand, binding to receptor. This can be an internal or an external receptor. You get then signal transduction, where you get different molecules signaling to one another, and then you get a cellular response. In the case of epinephrine, that is the breakdown of glycogen to glucose. There are different types of ligands. You can be small hydrophobic, nonpolar ligands. These pass directly through the plasma membrane because they're small and nonpolar. These are like nitric oxide, CO2, gases, very, very small molecules, and steroid hormones. And then we have ligands that bind on the outside of cell to receptors, membrane-bound receptors. These are water-soluble, polar, large, usually proteins. So intracellular receptors will bind to those ligands that are small hydrophobic ones. And these often bind the DNA. These, a big example of this are called transcription factors, right? Transcription factors are usually signaled through intracellular receptors. They go straight into the nucleus, binding the DNA, causing the transcription of necessary proteins, or mRNA and then proteins. And then we have cell surface receptors, which are found on the plasma membrane. 3 main kinds: ligand-gated ion channels, G-protein coupled receptors, and tyrosine receptor kinases. Ligand-gated ion channels are cell surface receptors that have a membrane-spanning region. And when a signaling molecule binds, it causes the channel to open and ions can flow through. Remember, ions are flowing down their concentration gradient, so they can either go in or out depending on that concentration gradient. G protein coupled receptors are one of the most common proteins that are targeted by pharmaceutical companies because they have a very clear structure with 7 transmembrane domains. G protein coupled receptors are always membrane bound. They are coupled to G proteins. G proteins are heterotrimeric, meaning they have an alpha, beta, and gamma subunit. When a ligand binds to the G protein coupled receptor, that causes a conformational change in the associated G protein that causes the G protein in the alpha subunit to bind GTP instead of GDP, which then elicits a cellular response or signaling downstream of that. Epinephrine is an example of a ligand that responds to a G protein coupled receptor. The third kind is a receptor tyrosine kinase. This is a 2-part receptor linked to kinase activity. And remember, kinases are responsible for phosphorylation. Receptor tyrosine kinases often respond to growth factors. So when you have a ligand bind, the 2-part receptor come together and they phosphorylate each other's tails through this kinase activity. Signal transduction pathways are chains of molecules that relay signals within a cell. Ligand binding initiates that. You get ligand binding, receptor changing shape, series of signaling events, and then a cellular response. And remember, signals always flow from upstream to downstream. So ligand being upstream, cellular response being downstream. Molecules, again, can be turned on or off through phosphorylation and GTPases. A good example of a full cascade is called the MAP kinase signaling cascade. Here, this involves growth factor signaling. From just looking at the receptor, you should know that that is a receptor tyrosine kinase based on the 2-part receptor and the phosphorylations on these tails. This signals downstream to MAP kinase, which acts, shockingly, as a kinase to phosphorylate the next thing. The MAP kinase then phosphorylates the next thing. This MAP kinase will then go into the nucleus to trigger the transcription of different genes resulting or necessary for cell growth. Second messengers are non-protein molecules that pass along a signal after the receptor binding. So there's 3 kinds. Calcium ions that get released from the ER can act as second messengers. Cyclic AMP or cyclic AMP, CAMP, often is downstream and is activated by a protein called adenylyl cyclase. These are both membrane bound. Ligand binds receptor changes conformational shape, activating adenylyl cyclase, which turns on CAMP, which then can act as a second messenger to turn on other things. We also have inositol phosphatases, which are membrane bound, and they can cause the cleavage of a membrane bound protein to become more intracellular. Second messengers are really important for amplifying signals. Signal transduction can be very, very complicated and can have different responses depending on the cell type. So cells are constantly computing different signaling pathways from the outside of the cell to determine what your cellular response will be. And even if you have the same ligand, in this case acetylcholine, depending on which cells have the receptor, you can get different responses. So in skeletal muscle, acetylcholine promotes muscle contraction via a ligand-gated ion channel, and in heart muscles, acetylcholine inhibits contraction through a G protein coupled receptor. Cellular responses can be at the molecular level, meaning transcriptional changes. And when I'm talking about transcriptional changes, I'm talking about turning a protein on, meaning you are turning a gene on rather, meaning you are translating DNA. That once you make mRNA, then that can make protein. So turning things on at the molecular level, meaning you are going to be making proteins, and this can also be at the macroscopic level, meaning cell growth or cell death, right? So gene expression is targeting transcription and translation to change the amount of proteins. Cellular metabolism, you are making enzymes more or less active in a cell. So there's lots of different examples here. We went over cholera and pertussis. These are both examples that work that are diseases that are caused in a similar pathway depending on whether you have an inhibitory hormone or a stimulatory hormone. And they result in different levels of CAMP and different signals. So there's lots of different questions to think about. What I think you should know about signaling cascades are you should be able to recognize GTPs, kinases, phosphatases. You should know that there's amplification. You should know there's ligands binding to receptors, and there's different kinds of ligands and different kinds of receptors. Just a little bit about development here. So when we go from zygote to embryos, we have a lot of what is called differentiation of those cells. And those cells have to turn into different types. So for example, one of your take-home questions was about formation of a neural tube. And you guys learned a little bit about how cells kind of fold into each other to create this neural tube. Individual cells, once you get an embryo, have to have different types of identities, neurons, intestinal cells, etc. These sources are based on intrinsic information. So what is in that original cell when the cell divides? You can have signals that are kind of partitioned to different parts of a cell. So when you get cell division, you get different fates for those cells. You can also have extrinsic information, meaning you get signals from other cells, right? We also went over embryonic stem cells, which are totipotent, meaning they can give rise to cell types of the entire body. We also have stem cells, which are pluripotent, give rise to many types, and differentiated cells, which are fully specialized. Differentiated cells, what makes them different is not differences in the DNA. Rather, that is caused by differences in mRNA expression. What genes have been turned on, meaning transcribed, meaning which proteins will be made through translation. That's what makes cells different, not the DNA. The DNA should be identical. Rather, it's what proteins have been expressed through transcription and translation.